What if the future of cancer treatment isn't chemotherapy alone, but programmable immune cells engineered like software? In this episode of Build with Shreya, I sat down with Henry Erdlei, doctor, scientist, and founder of GoCART, a biotech startup building next-generation CAR T-cell therapies for cancers like AML and multiple myeloma. But what makes this conversation fascinating isn't just the science, it's the realization that medicine is slowly becoming an engineering problem. Henry explains how CAR T therapy works almost like biological programming. Doctors extract immune cells from a patient, genetically engineer them to recognize cancer again, and send them back into the body to hunt tumors. It sounds futuristic because it is. But what stood out to me was this shift from generalized treatment to precision guided medicine. One of the most important insights from Henry was that modern cell therapies are powerful, but still limited by target selection. CAR T therapies work by identifying antigens, proteins sitting on the surface of cancer cells.
Yes. So by now we got different regimens for two kinds of antigen. We got CD19 and BCMA CARs. So CD19 CARs are against the antigen CD19 and are used originally in ALL, but currently they're mostly used in diffuse large B-cell lymphoma and can be used in diverse B-cell lymphoma classes. And then we have the BCMA CAR, which is used in the multiple myeloma. So what actually does GoCART do? If you look at these antigens, they are really specific, and they are chosen for a reason because they are so-called druggable antigens. So the problem with CAR T cells is you have to choose a target antigen, so-called an antigen is a surface marker, a protein on the surface of the tumor cell that identifies a tumor cell, but is not on a healthy tissue you desperately need to live. So, for example, the BCMA CAR is against this BCMA antigen on the multiple myeloma, but this is actually maybe the second or third best antigen on this type of tumor. Mm-hmm. Way better would be an antigen called CD56. And this is hugely expressed and it can't be downregulated like BCMA because it's one of the big growth driver of this type of tumor. So the question is, why don't we have this as a CAR T cell instead, or at least in addition to BCMA? And this, it is not druggable. Why is it not druggable? It is on the multiple myeloma and also on other types of tumors like the AML, mm-hmm. and glioblastoma, small cell lung carcinoma. But it's also on every neuron in your brain.
Okay.
And killing off all those is kind of counterproductive.
Absolutely, yeah. This is one of the core realities of biotech innovation. The problem isn't always whether we can destroy cancer, it's whether we can do it safely enough for the patient to survive the treatment itself. The most technically fascinating part of the conversation was Henry explaining GoCART's AND gate architecture.
And what we do is a so-called AND gate. Mm-hmm. While killing everything that has CD56 is pretty deadly, we might use it in a combination. So, for example, the AML, acute myeloid leukemia, has high levels of CD56, mm-hmm. and also expresses CD33, mm-hmm. a blood cell specific antigen. And in nature, they're not together. They're not together on the same cell, but they are together on this aberrant on the tumor cell. So what we want is a CAR T cell that kills off a cell only if it has both those antigens and not only one, the so-called mm-hmm. AND gate. So a bit similar to an AND gate in coding. You need both signal to get the reaction. And if you can manage that, then you can selectively kill off the tumor and nothing else. And this is also important safety layer for existing antigens, because like they come at a trade-off. So you kill off specific types of B cells when killing off everything that has CD19, or the right plasma cells if you kill everything with BCMA. So there are some downsides. It's just not so tragic that you wouldn't do it to get rid of cancer. But even those would profit from this kind of mechanism.
Henry's insight here is subtle but important. This is where biotech starts looking less like chemistry and more like software architecture. Henry also explains why CAR T therapies have worked better in blood cancers than in solid tumors.
There is, on one side, the challenge of the tumor microenvironment, especially within solid tumors. So if we look at these cell-based immunotherapies, by now we got approved regimens for hematologic tumors, diverse sets of B-cell lymphomas, the ALL, and the multiple myeloma have approved CAR T-cell therapy. It is kind of difficult to get into solid tumors. Why is that? Solid tumors grow a specific set of cells around them. They create kind of a tumor specialized organ, the tumor microenvironment, where they change their surroundings to benefit the tumor cells, which often includes immunosuppressive cells and agents being around there, and the whole environment being hypoxic, which is also suppressive to immune cells. So therefore, it's kind of difficult to get into there with the cell therapy and be really effective, unless you use tweaks to make the immune cells resistant to that or even activate by that. So there is approaches like hypoxia switches that can make them more alive, make them more aggressive inside this hypoxic environment, where you turn the hypoxia signal actually into a proliferation and not a stop signal. And also there is regulatory T cells the tumor attracts that tune down immune response via specific pathways. But you can also interfere with that and try to block these tricks in recent trials with CAR T cells. So we're getting there within the last steps.
What stood out here was the idea that tumors are not passive diseases. They adapt, they defend themselves, they evolve against treatment pressure. And because of that, modern cancer therapy is increasingly becoming a systems engineering challenge, not just killing cancer cells, but redesigning the environment around them. One of my favorite sections of the episode was when Henry explained how AI is already central to biotech research, just not in the way most people imagine. When people hear AI, they think of ChatGPT or image generators. But in biotech, AI is being used to predict protein structures, simulate molecular interactions, and optimize therapeutic designs before they even enter a laboratory.
So actually, technology is already pivotal in our process. The trimeric part we are building in silico actually is kind of an iterative AI process. So we start from a rather unspecific primer and have specific constraints, and then we train a model that predicts binding. And we have targets of these binding interactions, AB first, ABC then, and no AC or BC, because that would be a single binder binding to the CAR, which of course is what we want to avoid. The model learns this, so it iterates through, spits out mutated strands, and then ranks them. So we get a new list of antigens, trimeric parts, and the best performers of this mutated version, then they are mutated again. And it selects again the best performers. And this is basically how the in silico process for us works. So AI already is pivotal to create these artificial binding domains, which would be very hard in the pre-AI era, actually. So without, we don't use AlphaFold, we use a commercially available variant of that, of course. But it would be very hard to design this kind of peptide in this way without engines like that. And it would take probably many years to do this by hand.
This was one of the biggest mindset shifts for me. AI is not just automating communication anymore, it's helping humans design biology itself. A major theme throughout the episode was the sheer difficulty of building in biotech. Unlike software startups where you can ship quickly and iterate weekly, biotech founders operate under years of clinical trials, regulatory approvals, lab validation, manufacturing constraints, and ethical responsibility.
Of course, in medicine, it's kind of like the maybe a bit more demanding game you have to play, and you have to want to do this on the other hand. But the other hand, I think like motivation is pretty much there. You see, like when I think about every day new medical innovation is stalled, patients might suffer and patients do die in that case. So what better purpose can there be in a business? So actually, how helping a lot of people and maybe making a bit of money in the process. What better motivations can there be? And actually, well, by being a medical doctor, and that means I have seen the other side. So I have seen how the patients look and what kind of people are there and what is the customer, and that this is something that is really needed and that could make many people's lives way better.
So, if you were to give a piece of advice to entrepreneurs who are newly entering the field of cell therapies, what would be your advice and what is it that they should be expecting in the first few years?
My advice would be persistence and real data backing, because cell and gene therapies are highly regulated, even more regulated from the production perspective than traditional small molecule therapies, as they are kind of new, and many regulators, they consider them kind of obscure. So to ease them, you really need to get production and your data right from the start to avoid pitfalls of you need this experiment, we want more of this, and this can stall your venture by quite a lot. So what you should do is get neat data, get good assays that prove efficacy, already plan with assays for plasma half-life and some kind of safety assay before applying for clinical study to avoid getting set back. Probably will still get set back by regulators, but you can reduce that to a minimum by looking at what kind of experiments are kind of common for your type of therapy. For example, killing experiments in vitro where you culture target cells, so tumor cell lines, together with the CAR T cells is a common assay for CAR T cells.
This conversation felt like a reminder that some of the most important founders in the world are not building social apps. They're building therapies patients may someday depend on to survive. One line from Henry stayed with me throughout the conversation. Every day medical innovation is stalled, patients suffer, and patients die. That sentence captures the emotional core of this episode because beneath all the technical discussions around antigens, AI, and cell engineering, This conversation is really about urgency. If this episode changed how you think about cancer treatment, AI, or the future of biotechnology, share it with one founder, researcher, doctor, or engineer who believes technology should solve real human problems. And tell me in the comments, what fascinated you more?